Oil palm is one of the most important plantation crops in Malaysia. In 2018, Malaysia planted 5.85 million hectares of oil palm, covering more than 60% of its agricultural land. Previous study shows export revenue of oil palm in Malaysia is about 61.4 billion ringgit. However, the oil palm faces the threat of a devastating disease caused by pathogenic fungus, Ganoderma boninense. G. boninense is a white rot fungus which rots and kills oil palm trees. The basal stem rot (BSR) and upper stem rot (USR) diseases lead to the severe decline of the yield and shorten the productive life of oil palm. The estimated yield loss due to BSR is up to 1.5 billion ringgit a year. University Putra Malaysia (UPM) has proposed a solution using nanotechnology for the effective and efficient detection and control of the pathogen under the NanoMITe research grant, which is worth 5 million ringgit for 5 years. The program leader is Professor Dr. Nur Azhar Yusof from the Institute of Advanced Technology, ITMA UPM. The main objective of this research project is to develop a complete solution for detection and control of Ganoderma boninense problem through smart farming, employing nanosensors and smart nanodelivery system. In general, Project 1 focuses on the study of specific markers for detection of G. boninense based on secondary metabolites, elicitors, and protein expressed upon infection, and the development of nanosensor for early detection of this pathogen in the host. This project is divided into 4 sub-projects. The first sub-project develops biomarkers for early detection of G. boninense infection in oil palm. Root samples are harvested from the base of the palm trees. These trees do not have any basal stem rot symptoms. Ribonucleic acid (RNA) is extracted from the oil palm root samples. Polymerase chain reaction (PCR) is then carried out using specific PCR primers. This enables detection of biomarker genes at early stage of infection to enable effective control measures to protect field palms. This sub-project focuses on the hypersensitive response HR-inducing candidate elicitor proteins secreted by G. boninense strain PER71, the causal agent of BSR disease, purified using optimized anion exchange chromatography, SDS page gel electrophoresis, and size exclusion chromatography. Protein fractions collected from chromatography were tested for HR on oil palm leaves. Proteins in fractions that gave positive HR were identified using liquid chromatography mass spectrometry, LC, MS, and bioinformatics analysis. From the obtained sequences, 3 candidate proteins related to virulence, pathogenesis have been identified related to hydrolase enzymes that play a vital role in the process of infection on the host plant. The next sub-project aimed on the interaction of the biomarker with different transducer, i.e. optical and electrochemical, for detection of G. boninense at early stage in oil palm. The transducer sensor was then optimized for the best signal transduction. Electrochemical sensors have been developed using 2 different techniques, i.e. destructive and non-destructive, which applied voltammetry technique and electrical conductivity. Another non-destructive sensor developed in this study was electronic nose, e-nose. An e-nose is a mechanism that mimics on the same principle of human sensor smell. When humans breathe, all the volatile compounds that is suspended in the air will be detected by olfactory receptor and sends the information about the compounds to the brain, which will identify as a particular smell. For e-nose, the volatiles compound will be detected by an array of chemical sensors. The sensors generate a time-dependent electrical signal in response to the interaction of an odor on the sensor itself. Pattern recognition engine, which is equivalent to the human brain, will discriminate the odor based on signal received. eNose consists of several metal oxide semiconductor gas sensors in a chamber box. The MOX stick used in this study is a USB interface for excitation and readout of resistive sensors. This subproject aims on identification of secondary metabolites present at early stage of G. boninense infection, was employed using nuclear magnetic resonance (NMR) and gas chromatography mass spectrometry GCMS-based metabolomics. Oil palm seedlings treated with G. boninense inoculated rubber woodblocks were used in this study. Leaves and roots of healthy and G. boninense infected oil palm were extracted using 80% methanol. The crude extracts obtained were analyzed using NMR and GCMS. From the analysis, level of secondary metabolites such as phenolics, steroidal compounds, and fatty acid derivatives were found more abundantly in infected samples compared to healthy samples. Project 2 studies on the ineffectiveness of Ganoderma treatment due to the failure in delivering the fungicide to the disease location. The development of nanocarriers of agrochemicals with targeted delivery and controlled release capabilities have lately attracted considerable attention. Thus, our research work aims to formulate an effective delivery of fungicide by the encapsulation of fungicide onto a nanocarrier as the host matrix. The formulation of fungicide nanodelivery system aims to reduce the fungicide toxicity, enhance the efficient delivery of active ingredient to the target site, able to sustain in a longer time, and consequently improve the fungicide efficacy. Therefore, the findings of this study will help to sustain the economic benefits of the palm oil industry by controlling the basal stem rot diseases caused by Ganoderma through fungicide nanodelivery system. The formulated system has low toxicity, hence makes it harmless to the planters and person who will be applying it. The controlled release properties will decrease the fungicide wastage and leaching. Thus, environmental issues such as runoff and accumulation of fungicides will be greatly minimized. In addition, the long circulation time would overcome the shortage of workers in the oil palm industry by reducing the frequency of applying time in the infected tree. As a conclusion, this new formulation of agro-nanochemical is a promising candidate to be used for Ganoderma treatment. Project 3 has found a certain method of detecting the specific DNA of G. boninense as an initial stage for agricultural diagnosis. The tool is Lab-on-Chip. This device comprises of two components, biosensor and microfluidic. In this project, a microfluidics chip is fabricated in a way that once a raw sample of the oil palm, preferably root, is processed into liquid and flowed through the inlet of the microfluidics chip, DNA extraction occurs in the channels of the chip. The end product, which is the target DNA, will meet a biosensor which is immobilized with probe DNA specific to Ganoderma boninense. Project 4 focuses on the implementation of the nanodelivery system in smart farming for actual detection and control of Ganoderma boninense. Project 4 consists of 3 sub-projects. The first sub-project aimed at developing a new mechanical system that is able to deliver the treatment solution effectively and efficiently. The entire system is called the drill-jector. The innovative concept developed here is on the integration of drilling and injecting, which can be done in one phase. In addition, the driller is made to be the smallest possible to reduce damage to the tree. The rate of pressure and flow can be controlled. The solution can be delivered as a liquid form or nanogranular size. The drill jector can be operated as a hand tool or reconfigured as an actuator to a larger machine. In the next sub-project, nanoporous pipe (NPP) for irrigation and drainage purposes were developed to cure BSR disease by killing Ganoderma boninense when submerged for 3 months and above. Several prototypes of aluminosilicate base mixed with other forming agents were developed and tested with SEM-TEM to determine pore distribution for better water uniformity. NPP is then characterized for its hydraulic properties, while soil wetting pattern is monitored in a cubical soil column to observe the wetting dimensions when placed beneath soil surface. Engineers can utilize information on water flow over time through the soil media for good irrigation water supply design. With installation of soil moisture sensor, water delivery through NPP can operate automatically by receiving signal from microcontroller to stop a pump when soil moisture reach a desired level. This system was applied at oil palm nursery to monitor the effectiveness in curing BSR and found that continuous supply at saturation level for a long period can increase the mortality rate of Ganoderma boninense. Therefore, BSR can be eliminated and the oil palm production is maintained. The last sub-project studies on the development of MyGano Online. It is an online system which can provide a real-time monitoring and alert systems in detection and monitoring the oil palm tree infected by G. boninense. It is developed by using Internet of Things (IoT) and remote sensing technologies. The system consists of 3 subsystems which presents different approach of detection, namely soil sensor network system, laser scanning canopy biometric system, and spectral properties system. The soil sensor network system SSNS detects the BSR disease by monitoring the condition of soil properties. Data taken by the sensors were transferred from the field to the cloud by using the 3rd generation 3G cellular connection. Users can view the data in the web application and receive alert notifications if the data exceeds a certain value. Laser Scanning Canopy Biometric System LSCBS evaluates the severity level of G. boninense infection using point cloud data. 3D image and physical descriptions of oil palm were obtained using terrestrial laser scanner TLS. The users can key in the data obtained by TLS to check the health status of oil palm. The users also can view the location of the trees and retrieve the parameters data developed using the ArcGIS online application. Spectral Properties Detection System SPS use spectral approach to detect G. boninense. Thermal images of canopy were captured at 3 different angles. The users will be able to see the healthiness status of the oil palm tree in a map and individually by looking up to the tree ID. All systems used machine learning techniques to classify between healthy and unhealthy tree. Furthermore, the laser scanning canopy biometric system can provide detection at different severity levels of infection.